Hello, General Biology, and welcome to the third in the screencast series of cellular respiration. And we're going to start this one off with the amoeba sisters. So here we go.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it takes a while for me to feel like I get my energy back. It takes a lot of time and coffee for that to happen for me. Cells don't really have that luxury. They're busy performing cell processes all the time, and many of the processes that they do require energy, specifically ATP energy. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it's action-packed with 3 phosphates. When the chemical bond that holds that third phosphate is broken, it releases a great amount of energy. It also is converted into ADP, adenosine diphosphate. And really, that's just a fancy way of saying that it has 2 phosphates after losing 1. So where am I going with this? Well, cells have to make ATP energy. It doesn't really matter what kind of cell you are, prokaryote or eukaryote, you have to make ATP energy. The process for making that ATP energy can be different depending on the type of cell, but you have to make ATP energy. One way that this can be done efficiently is called aerobic cellular respiration. We are going to focus on aerobic cellular respiration in eukaryote cells. Remember that eukaryote cells have many membrane-bound organelles, such as mitochondria. They're going to be kind of a big deal in this. So let's get started. Remember, we are trying to make ATP energy. Let's take a look at this formula. Remember that reactants, the inputs, are on the left side of the arrow, and products, the outputs, are on the right side of the arrow. This formula, by the way, looks remarkably similar to photosynthesis. I mean, just look how the reactants and the products just seem to be on different sides. This isn't really a coincidence. See, in photosynthesis, organisms like plants and some types of protists, for example, they make glucose. Notice how glucose is a product. But in cellular respiration, we break the glucose. Notice how glucose is a reactant in order to make ATP energy. So, photosynthesis makes glucose, and cellular respiration, it breaks glucose. Kind of cool. Now, photosynthetic organisms, they have the best of both worlds because really, they don't just get to do photosynthesis to make their glucose, they also get to do cellular respiration to break their glucose. I say that's pretty great because glucose is the starter molecule in cellular respiration, and it's needed in order to get all of this going. So if you aren't photosynthetic, like a human or an amoeba, you have to find a food source to get your glucose. Now, cellular respiration involves 3 major steps. We are going to assume that we are starting with 1 glucose molecule so that you can see what is produced from this 1 glucose molecule. Now, we're going to start with the first thing. Number 1, glycolysis. This first step takes place in the cytoplasm, and this step does not require any oxygen. Glucose, which is the sugar from the formula, is converted into a more usable form called pyruvate. It takes a little bit of ATP energy itself to get this process started. The net yield from this step is approximately 2 ATP molecules and 2 NADH molecules. What is NADH? Well, NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on, and we'll get to that in just a little bit. Okay, number 2 is the Krebs cycle. This is also called the citric acid cycle. We are now involved in the mitochondria, and this step requires oxygen. The pyruvate that was made before, it's now converted and it will be oxidized. Carbon dioxide, CO2, is produced, and we produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like the NADH, and it will also assist in transferring electrons to make even more ATP. The third step is the electron transport chain. This is just a beautiful thing. We're still in the mitochondria, and we do require oxygen for this step. This is a very complicated process, and we are greatly simplifying it and saying that electrons are transferred from the NADH and the FADH to several electron carriers, and they're used to create a proton gradient. The protons are used to power this amazing enzyme called ATP synthase. Really, if I could be any enzyme, I'd be ATP synthase, because this enzyme takes phosphates and adds them to ADP. And if you add one phosphate to ADP, you make ATP. So that's what ATP synthase does. It makes ATP, and it makes the ATP by adding those phosphates. Now, oxygen, it's the final acceptor of these electrons, and when you have an oxygen molecule combining with two protons, you get H2O, which is water. So water is a product as well. The electron transport chain produces a lot of ATP compared to the other two steps. It's all because of that ATP synthase. There isn't an exact number on this. Many textbooks will say that this step makes 34 ATP, meaning that the net amount of ATP when you added all three steps would be 38 ATP. But you need to understand that this is a perfect case scenario. And in general, you can't really expect that much ATP made each time. Now, if we look at our formula again, we can see how the glucose and the oxygen on the reactant side were used to produce carbon dioxide, a waste product, water, which was a waste product, and ATP energy. ATP energy was our goal. Now, this was just one way of creating ATP energy, a very efficient way at that. But like we had said at the beginning, all cells have to make ATP energy. The way they do it, though, can differ. If there is no oxygen available, some cells have the ability to perform a process known as fermentation. It's not nearly as efficient, but hey, it makes ATP when there's no oxygen. Now, we really can't emphasize enough how important the process of making ATP energy is. If you doubt how powerful it is, consider cyanide. This toxin is found in some rat poisons. It is highly toxic. It works by blocking a step in the electron transport chain, and without being able to continue the electron transport chain, cells can't produce their ATP, and this poison is deadly in a very short time frame. There is also a demand for increased research on various mitochondrial disorders. Mitochondria are so essential for ATP production in humans that many mitochondrial disorders can be deadly. We are confident that the understanding of how to treat these disorders will continue to improve as more people, like you, ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, now that you saw that, let's take a look at the reactions of glycolysis. So we're going to look here, and we're going to see we have a 6-carbon compound, and this 6-carbon compound is glucose. This is our sugar. And remember the chemical formula. Let me see if I can write here again. I know writing is difficult. We're going to have C6H12 and then O6. Now, even though we're just showing the carbons here, the hydrogen and the oxygen are still present. But it's going to be important that we know that this is an endergonic molecule, and we need to break it down to get ATP. So in order to break this down, we need ATP. We have to have ATP present to break down this glucose molecule. And remember, glycolysis, this is happening in the cytoplasm. So here's our ATP. This ATP is going to break. Remember, if I draw ATP, here's A, and then I had a high energy bond with a phosphate. This is one phosphate, and a high energy bond, and another phosphate, and a high energy bond, and another phosphate. Let's see if I can change colors here. Let's go for blue. Alright, so what's going to happen is, in order for this ATP to be able to break down this glucose molecule, that means that the bonds between the second and the third phosphate group need to break. Now to break down this glucose molecule, we're going to need 2 ATPs to do what I've described down here. When it does, we're going to wind up getting ADP and an independent phosphate group. Now in this diagram, they didn't draw this independent phosphate group in there, so you're just going to have to automatically assume that it's there, but it is there. So 2 ATP were invested to break down this glucose molecule. And with the releasing of energy that comes from this, that energy will be used, to break down, this is so hard to write with, to break down that glucose molecule. And we'll get 2 3-carbon compounds from this. Now, we've spent these two. We're not going to worry about those for right now, but we're going to focus on these 2 3-carbon compounds. So what happens is, when we break these up, you know, hydrogen only has 1 electron to it total. It only has 1. And what happens is that electron gets ripped away from the hydrogen. So 2 of the hydrogen, I should say actually 4 of the hydrogen, 2 for each of these 3-carbon compounds, get pulled away from the hydrogens. So we have a carrier compound called NAD+. You heard the amoeba sister say it's a coenzyme. We know coenzymes are helper enzymes, so this is going to help with the reactions that take place. Notice that this has a plus sign to it. Electrons are negative. I didn't draw the negative signs in here, but we know that they're negative. So 2 of these electrons are going to go to 1 of the NAD pluses. The other 2 of these electrons are going to go to the other, and when they do, they're going to make something called NADH. And this NADH is going to go to the electron transport chain. All right, how are we doing so far? Take a deep breath. Write down. You'll notice on your paper that you've got places to write notes, and maybe you could write a question there if you don't understand something, so that you can ask it in class. All right, so whenever we pull, and I'm going to switch colors again. Let's do this. Let's try a green. Whenever we pull these electrons away from these 3-carbon compounds, energy is released. So this is energy again. Oops, oh boy, I'm really ruining this one up. Energy. Hopefully you'll be able to read it. It's not easy. And that energy is going to be used to join ADP with those independent phosphates that we lost here. As a matter of fact, there's enough energy for 4 ADP to be restored back into 4 ATP. Okay, so we get a total of 4 ATP, but remember we invested 2, so we're only really gaining 2 ATP from the process of glycolysis. The remaining 3-carbon compound that we have here is pyruvic acid, or a pyruvate. Whoops, I'm running out of room. That's an IC, so pyruvic acid. All right, so that is the process of glycolysis. I'm looking at our time, and we're running out of time, and we'll pick up from here before we start into the next cycle. See you in a few.